Hey guys, how are you? I hope you're doing well. We are going to talk about metabolism and nutrition today. We're going to talk about the processes that are involved in anabolic and catabolic reactions. We're going to list and describe the steps necessary for carb, lipid, and protein metabolism. We're going to talk about the processes that regulate glucose levels during the absorptive and post-absorptive states. We're going to talk about how metabolism is essential to maintaining body temperature, like thermoregulation, and summarize the importance of vitamins and minerals within the diet. So remember, metabolism is the sum of these catabolism and anabolism, right? So catabolism is breaking down those molecules. Cats like to break things down. And anabolism is the synthesizing of new molecules, right? And so we do these catabolic reactions. And then we can also do anabolic reactions, but you can see right here in the center is glucose, right? And so the goal is to kind of get this energy, right? And so this is sort of our energy line, okay? So we can take carbohydrates, we can break them down into simple sugars, which would be like glucose. That glucose can then enter into glycolysis. It can form pyruvate, which is then going to form acetylCoA, which can go into our citric acid cycle. Okay, and then we can get some substrates for amino acid synthesis, and we can also go in and get our energy out of that. Right, that is cellular respiration right here. Okay. And so we can take our fats, we can break them down into glycerol and fatty acids, and they can plug in to this area of cellular respiration. Okay. We also can take proteins and we can break them down into smaller particles, and we can again plug them in here so that we can do cellular respiration. We can also, if we have extra glucose, we can store it as glycogen. And storing is our anabolic reaction, right? So we're going to build up that glucose. We're going to combine glucose plus glucose plus glucose plus glucose. Remember, it's going to form glycogen, okay? We can take our fats, right? And so they can come in here and we can store them as fatty acids like phospholipids and fats. Okay, we can take our proteins and we can store them as our amino acids. Okay, so we store them as our muscle tissue typically, right? So our catabolic reactions, when we break things down, we get the energy out of the bonds. Remember, the bonds are like the glue, and the glue is that energy. Okay, and then anabolic reactions are where we store energy. So we have to add glue in, and so we're adding in the energy. There's different types of hormones that are going to allow us to break down. Okay, so there is cortisol. Cortisol, if you remember, functions it's released from the adrenal gland, and we release it in response to stress. Okay, its main role is to increase our blood glucose levels, and it does that by doing gluconeogenesis. Okay, gluconeogenesis, remember, we break down fats and proteins. Okay, and when we break down those fats and protein and then put it in and form new glucose, right? So gluconeogenesis is done by being both a catabolic and an anabolic, okay? Then glucagon, okay? So glucagon is released from our alpha cells in our pancreas, right? And they do that when we are in our starvation mode or when the body needs to generate some extra energy. Okay. And so it's going to stimulate the breakdown of glycogen in the liver to increase our blood glucose levels. Adrenaline or epinephrine are released in response to our sympathetic nervous system, and they also are going to stimulate gluconeogenesis. Now remember, gluconeogenesis is building up, but in order to build up the glucose, we have to first break down the fats and the proteins. I hope that makes sense. And so that's why we have them as being catabolic first. Okay? And then for anabolic hormones, this is where we're going to be synthesizing, okay? And so anabolic hormones are like growth hormones. Growth hormones stimulate the growth of cells and tissues. Insulin-like growth factor is going to stimulate the growth of muscles and bones. Insulin is produced by the beta cells in the pancreas, remember? And they do this in response to having a higher glucose level because we've just eaten. and they take up the glucose from the blood and store it in the liver and the muscles as glycogen. Okay. Testosterone produced by the testes in males and the ovaries in females is going to stimulate an increase in muscle mass. And then estrogen, which is primarily produced by the ovaries, but it's also produced by the liver and the adrenal glands, is going to increase the metabolism and the fat deposition. Okay. So remember, cellular respiration is when we take glucose in the presence of oxygen, we get out carbon dioxide, water, and energy. Okay. There's 4 steps to cellular respiration. There's glycolysis. Then there's some sort of transition reaction. This is where we're changing something. The Krebs cycle, and then the electron transport chain. Okay. So for carbohydrates, again, are organic molecules. They're composed of carbon, that's the carbo part, hydrogen, high, and then the oxygen is the O in there, okay, carbohydrate atoms. And those are, we can have them as monosaccharides, so those are simple sugars like glucose and fructose, or we can have them be complex sugars like disaccharides and polysaccharides. These are going to be like starch or glycogen. This is our storage molecule. Okay. Carbohydrate digestion is going to begin in the mouth. Remember, we talked about salivary amylase when we talked about the digestive system. Okay. And so it starts within the mouth. You can actually take a piece of bread and put it in your mouth. Just let it sit on your tongue for a second, and it's going to start to taste sweet. Okay. And the reason that it's starting to taste sweet is because the starches that are in the bread are being broken down by amylase, which is an enzyme, right? That ASE at the end tells us it's an enzyme. And so that's going to allow those monosaccharides, those simple sugars, to be absorbed across the epithelium. Okay. So this is glycolysis. So once we absorb those monosaccharides, they get transported into the tissues. And then we can start that process of cellular respiration. And the first step of cellular respiration is glycolysis. Okay. And so with glycolysis, the important part here is that we start with 1 glucose molecule. That's then going to get us 2 NADH and 4 ATPs. Now, in order to start this, we had to put in 2 ATPs. So the net is that we get 2, right? Because we got 4 here, but we had to put 2 in to start. So 4 minus 2 is 2. Okay. But the key really is that we're going to get out this NADH molecules. They're going to continue. And we're going to end with these 2 pyruvate molecules. Okay. Then we're going to take these pyruvate molecules and we're going to transfer them, this is that transferation reaction, into acetyl-CoA. Okay? That is acetyl coenzyme A. That acetyl-CoA is then going to go into this Krebs cycle. And when it goes into the Krebs cycle, it is going to produce these high energy molecules, NADH and FADH2. Okay, and so this is the cycle. And that means that we can put in different molecules throughout. Okay, so we like to start with acetyl-CoA, but we don't have to. Okay, and then what are we going to do if we have oxygen versus no oxygen? Okay, so the process of anaerobic respiration is when we convert glucose into 2 lactate molecules. We do this when we don't have any oxygen. So this would be anaerobic respiration, right? No oxygen, okay? And so when we do that anaerobic respiration, we're going to get lactate out, okay? And here's that. On this side, when we have our aerobic respiration, we're going to get all the way down to the bottom. So we're going to do this whole process of cellular respiration, okay? We do what's called oxidative phosphorylation. This is also called the electron transport chain. And this is the last step of cellular respiration. Remember, there were 4 steps. Glycolysis, okay? This is glycolysis right here, okay? The transition reaction, which is where we're converting that pyruvate into the acetyl-CoA. The citric acid or Krebs cycle, it's also called the TCA cycle, lots of different names, same thing, okay? And then the electron transport chain is our last step of cellular respiration. Now, this is when we have oxygen present, okay? So the electron transport chain is just a series of electron carriers, okay? And then ion pumps. They're used to pump these hydrogen ions out of the inner matrix of the mitochondrial. So here's the mitochondrial matrix. This is our intermembrane space, okay? And so we're pumping. and pumping. And we're creating a concentration gradient. We're concentrating this hydrogen molecules up here. And then we can shove through this molecule right here, okay, we're going to send the hydrogen ions through it, okay? And the hydrogen ions want to go through ATP synthase, which is what this molecule is right here, okay? They want to go there, right? Because they want to diffuse through it, because this is an area of high concentration. This is an area of low concentration. When we shoot the hydrogen ions through, they can then do what's called ATP synthesis, right? So we can take the ADP here, adenosine diphosphate. You can't see my mouse, there it is. And we can convert it to ATP, adenosine triphosphate. And remember, the energy is in that bond, okay? So this is sort of a summary for us, right? So this is glycolysis, our first step here. And out of glycolysis, we're going to get out those two NADH molecules, okay? Then we're going to do our transformation reaction where we're going to convert acetyl-CoA into, again, two NADH molecules. Then we're going to do the Krebs cycle, okay? And again, we're getting out 6 NADH molecules, but also the FADH2, okay? These are our electron carriers. They're also these really high energy molecules. They're going to go through that electron transport chain. And as we move through that electron transport chain, we're going to get out 34 ATP. We got out 2 ATP here. So look at that, 36 ATP total. Okay. So when we don't have oxygen present, that's this part. Okay. This is where we need the oxygen. When we don't have oxygen present, then we're going to form lactic acid molecules, right? We're going to get lactate instead. Okay. So we also we can do gluconeogenesis. Now, gluconeogenesis means new. That's what neo means, new. Genesis means to create. Okay. So create new glucose. Gluconeogenesis. Create new glucose. And that's exactly what we're doing. And we can synthesize new glucose molecules. We can do it from pyruvate. We can do it from lactate. We can do it from glycerol. That's from fatty acids, right? Or we can do it from amino acids, okay? So this is we can take the proteins, break them down, get those amino acids out, and then we can shove those amino acids back together. We can form glucose, okay? Same idea with glycerol. As glycerol comes from fatty acids, we can then make glucose from that glycerol. Okay, this is going to take place primarily within the liver during periods of low glucose. Okay, and certain key organs, including the brain, can also use only glucose as an energy source. This is up for debate still. So this used to be believed that it truly was just the brain could only use glucose, but now we're finding that maybe that's not true. Okay. So when we break down lipids or fats, okay, they're generally formed as triglycerides. Remember, triglycerides have the 3. That's what the tri means. This is a mono, these fatty acid chains attached to it, okay? And so lipid metabolism begins within the intestines. And there we ingest the triglycerides. They're broken down into smaller chain fatty acids. So we get them down to monoglycerides. Molecules by our pancreatic lipases, which are going to break down those fats, they emulsified by those bile salts. That means, remember, they break down, even smaller. Okay? The fatty acid molecules become free fatty acids and they travel across the intestinal membrane. Once they go across, they recombine to form those triglycerides again. We do that through these things called chylomicrons. Okay, so these are the triglycerides here, right? And they sort of these phospholipid bilayers. They enable the fats and the cholesterols to move within this aqueous environment of our lymphatic and our circulatory systems. We put this phospholipid head out, and then the tails are inside, right? Because the tails are hydrophobic, meaning they don't like water, and the heads are hydrophilic, meaning that they can live within the water or the aqueous environment. Lipolysis, okay, in the cytoplasm, this is where we break down the triglyceride even smaller. They get broken down into fatty acids and glycerol. Lysis means to break. Lipo means the fat. So they break down the fat. When the glucose levels are low, the triglycerides can then be converted into those acetyl-CoA molecules. They can then be used to generate ATP through that aerobic respiration. So they can plug in, okay, to the Krebs cycle. So we have that fatty acid CoA gets broken down. It can enter into that matrix, and we get that fatty acid CoA right here. And we can then enter into the Krebs cycle. Okay. Ketogenesis is when we have excessive acetyl-CoA. Okay. It gets created from the oxidation of fatty acids. The Krebs cycle can get overloaded and it won't be able to handle it. So then that acetyl-CoA gets diverted and it creates ketone bodies. You may have heard of ketone bodies. People actually try to put themselves into this idea of ketosis. Okay. So ketones are going to serve as the fuel in times of prolonged starvation when the patients suffer from either uncontrolled diabetes and they can't utilize most of that circulating glucose, or when they intentionally do this to themselves, meaning that they eat a high protein diet and a very minimal amounts of carbohydrates, so minimal amounts of glucose are being eaten. Okay, so the organs that have classically been thought to just be dependent on glucose, like the brain, can actually use those ketones as an alternative energy source, and that's new information. When glucose is limited, the ketone bodies can be oxidized, and they can also produce the acetyl-CoA and go into that Krebs cycle. Okay, so we can do that again with that excessive proteins and with the excessive fatty acids. We could put them into the Krebs cycle. Lipogenesis is where we're building up the fats back up, right? So when we have excessive acetyl-CoA generated by glycolysis, we can then convert it into fatty acids, triglycerides, cholesterol, steroids, and even bile salts. This happens within the cytoplasm of our fat cells and in the cytoplasm of our liver cells. When we eat more glucose or carbohydrates than our body needs, your body can use that acetyl-CoA to turn the excess into fat. Mommy. We're going to